Here today, let me remind you of the events at the center in the coming days. On Tuesday, there is no lunchtime talk, but we have Andrea Ludgers, who is going to give the first lecture in the annual lecture series. So the talk is at 3:30 on the 10th floor, followed at 5:15, 5:30, with a reception here at the center. And you're all invited to join this talk. And the talk will be about model templates and model-based unification. On Tuesday next week, we'll have another lunchtime talk given by our senior visiting fellow, Karim Khalifa, who is right there. And Karim will be talking about unifying sociological race theories. And on Friday next week, we're welcoming back Sherife Teki, who was a postdoctoral fellow here years ago, and she's been a fellow here at the center, and Sherife has been here very, very often, so we are actually delighted to welcome her back. If you're interested, we have two workshops coming. One in November, organized by Wenwu, right here on the philosophy of neuroscience. The program is on the center's website, and for the spring workshop, the workshop is called "To Be or Not to Be," included in the causal model. It's about the selection of causal variables. If you're interested, there is a call for papers on the center website as well. Today, it's my pleasure to introduce Goodwin, who is a fellow at the center this semester. He is a professor at the University of South Florida. He works on many different topics: the philosophy of math, the general philosophy of science, the philosophy of climate science. And he was one of the first, maybe probably not the first, one of the first to work with the philosophy of chemistry, when the philosophy of chemistry really wasn't a thing at all.
Still isn't.
It is a thing. It's not a cute thing. I think it's true for a very long time. Just no one was writing about chemistry. So he also has interest in the history of philosophy, the history of philosophy of science with Kant and Kuhn. He has published on all of these topics in the best journals, including the GPS, philosophy of science, and so on. We've been very lucky to have him at the center this semester.
Well, thank you. It's a pleasure and a privilege and a pleasure to be here. I was just thinking about the last 13 years of my life where I've been teaching biomedical ethics to semi-interested undergraduates and wondering about my career choices. But I'm glad to say that since I've gotten here to the center, I've sort of felt a rebirth of interest in philosophy, to be surrounded by a bunch of people who are interested in the same things and are smart. It's really helped reawaken my love for this discipline. So I'm really thankful to you guys for having me here and letting me give you this talk. And I look now at my title, and I think I'm sort of overselling in one half and underselling in another. So my interest in interpreting Kuhn is sort of more general than just talking about his speciation metaphor. Kuhn's one of those philosophers that the reactions to which is kind of bimodal. You sort of either love him or you hate him. And if you love him, you read him in a way where you're trying to vindicate him. And if you hate him, you're trying to burn them all down. I have a little bit more nuance the way I try to read him. I'm sort of a pro-structure guy, and he deteriorated after that sort of reader of Kuhn. And so what I'm really trying to do here is develop a sort of interpretive concept for thinking about Kuhn after structure. So you'll see that I kind of do this in this talk. And this is and I use the birth of biochemistry. Here's where I'm overselling. I'm really talking basically about Linus Pauling and his work as an emblem of sort of the birth of biochemistry. Biochemistry is too huge to talk about its birth in anything other than some minuscule slice of it. So I'm going to be talking about Pauling, so not really the birth of all of biochemistry. But I'm going to use that as an example to try and reflect on something that Kuhn introduces later in his career, what's called the speciation metaphor, and how to think about its relationship to earlier Kuhn. Okay, so I kind of do that through the work of Linus Pauling. That's the overall idea. Okay.
Press the button.
There we go. Okay. Well, it's on here, but
No, because I'm pushing back.
okay. Okay. All right. So the basic tool I think that we should use for understanding Kuhn's philosophical development is what I'm going to call incommensurability mission creep. So if you're not familiar with the term mission creep, it's a kind of description of what happened when you send soldiers into Afghanistan to capture Osama bin Laden. You capture them and then they're still there 20 years later, right? So somehow the mission of staying there evolves over time and reinforces engagement with the soldiers over time. So they killed him. They killed him and then they stayed. That's the point, is that the mission expanded over time. Okay, and so basically, I think that this is going on in Kuhn, and many times to his sort of detriment. So first quote I have here is Kuhn describing his introduction of incommensurability in structure, which I think is an accurate description. So the term incommensurability is a metaphor in structure. It comes from mathematics, talks about the relationship of a hypotenuse of an isosceles triangle to one of its sides. There's no common measure, but they can be compared. So it's a metaphor that's supposed to tell you about the relationship between two scientific practices, or in revolutionary science. They can be comparable without being comparable in a straightforward way. So that's where incommensurability gets introduced in structure. Yeah, and then this is him describing his last book. No other aspect of structure has concerned me so deeply in the 30 years since the book was written, and I emerged from those years feeling more strongly than ever that incommensurability has to be an essential component of any historical, developmental, or evolutionary kind of scientific knowledge. So it's gone from a metaphor to the central idea of his philosophical account. So I want to say the mission is creeped. Now there's some obvious cases where I think this happens, like the notion of revolution, but I'm not going to talk about revolution today. I'm going to look at another case of incommensurability mission creep in Kuhn, and particularly in his concept of scientific progress. Okay. So some of you remember that Kuhn had an early account of progress and structure in basically chapter 13, and he introduces what's sometimes called his first Darwinian metaphor for scientific progress. In revolutionary science, the community of scientists selects among the viable alternatives. And so over time, you get the community selecting and you get evolution from beginnings, but not towards the truth. The sort of first metaphor you get for evolutionary metaphor in Kuhn. And then many people who read structure saw incommensurability as being a threat to the authority of science, either because it undermined realism or rationalism. And so in response to this, Kuhn says, no, it's not a threat. It's the very foundation of the authority of science. Incommensurability is the key to understanding why science has authority. So it's like, you're criticizing me for this term. No, it's actually the key. So incommensurability, properly understood, can reveal the source of the cognitive bite and authority of the sciences. So it's gone from, we had the story of progress, he gets criticized for incommensurability, then incommensurability becomes the key to progress. How is incommensurability the key to scientific progress? He says its role as an isolating mechanism supports progress in science along two dimensions, both depth and breadth. So this idea that you should think about scientific progress on these two dimensions was already there in structure, but he develops it later. And it's just the idea that within a particular paradigm, you focus on narrower and narrower and more precise problem solving. And so you get more depth within paradigms and you get breadth by giving birth to more and more specialties, which examine different parts of the world. So science progresses in depth and in breadth. And he wants to say later on that incommensurability is crucial to both dimensions. Incommensurability is crucial to progress in breadth and progress in depth. All right. In order to do this, this notion of incommensurability changes somewhat in later Kuhn. It's not just a relationship between two practices in this, two ways of doing science within the same discipline, like, Newtonian optics versus wave theory of optics that are incommensurable. It's between adjacent disciplines. So, something like chemistry is incommensurable to physics. So it's a relationship between adjacent disciplines, not just within practices within the same discipline. So he broadens the notion of incommensurability when he gets to these later claims about how it functions as an isolating mechanism supporting depth and breadth of scientific progress. Okay. Okay, so I'm going to sort of break it down into two pieces. The first one is the breadth dimension. So what does he mean that incommensurability helps promote the proliferation of specialties? That's what he's trying to explain here. Well, he says scientific specialization is analogous to biological speciation. So biological speciation is the process by which a species splits into two, basically, right? So you have subpopulations that gradually evolve reproductive barriers, and then they can no longer breed, and they're distinct species. That's the process of speciation, and he wants to say that scientific specialization is like that. Except whereas a species is a reproductively isolated population, a specialty is, and then the quote up here, a community of intercommunicating specialists whose members share a lexicon that provides the basis for both the conduct and evaluation of the research, and which simultaneously, by barring full communication with those outside the group, maintains their isolation from practitioners of other specialties. So incommensurability isolates them communicatively from other specialists. And in that sense, it's analogous to a reproductive barrier, yes. All right, and so if specializations are like species, then revolutions are like speciation. They result in a new lexicon and thus communicative barriers between scientists. So revolutions, which produce new divisions between fields in scientific development, are like episodes of speciation, biological evolution. So what's the idea here? It's something like a sort of subgroup within a discipline has to tweak their concepts in order to solve a certain anomaly. And those concepts are sort of in conflict with the concepts of the baseline discipline, and eventually they sort of develop into distinct lexicons and distinct specialties. And so these incommensurable conflicts between concepts are the source of specialization. And then here's this is a quote from Kuhn describing this analogy. Over time, a diagram of the evolution of scientific fields, specialties, and subspecialties comes to look strikingly like a layman's diagram for a biological evolutionary tree. Each of these fields has a distinct lexicon, though the differences are local. There is no lingua franca capable of expressing the content of them all, or even of any pair. So each distinct subdiscipline is lexically separated from every other. This image. Well, I mean, there's a lot of follow-up questions you might ask about that. I'm just going to focus on one that's sort of useful for me later on. And that is like, it sure sounds when Kuhn talks about this, like he thinks that the route to specialization in science is via revolutions and incommensurable conceptual differences. But you might wonder, is that really necessary? I mean, couldn't we have specialization without incommensurability? I mean, when there are a sort of broadly sociological alternative, just people who are working on different parts of the world are going to develop tools that are adapted to different things, but not in any deep sense incommensurable. So think about something like organic chemists and macroeconomists, economists. Surely there are, communicative barriers between them. There's not a lot of intercourse between these two scientific fields. But again, you don't want to say that that's because they carve up the world into different objects. They're just talking about different parts of the world, right? And you might say that the lingua franca that would allow you both of them to talk together would just be the union of their conceptual schemes for their part of the world. Now, if that's right, if we can separate out specialties that have trouble talking to each other, because well, I guess I should back up. So this deep investigation requires specialization. Specialization requires narrow focus, and different foci make communication different. That's kind of the way Kuhn presents it in structure. So he talks about how when you adopt a paradigm, you get much more deep problem solving because you have all these assumptions in common with the other people you're working with, and you can do detailed investigations of nature. Nothing about incommensurability involved there. Okay, so the point is that I want to distinguish two questions. Are scientific specialties distinct and in restricted intercourse, like, say, organic chemistry and macroeconomics? And is the cause or the best explanation of this distinctness and restricted intercourse incommensurable conceptual differences? Those are two different questions, I think, and you might answer yes to this and no to that. Okay, and that makes space for the question, are there other plausible explanations of specialization and restricted communication than an appeal to incommensurable concepts? In other words, is non-revolutionary specialization possible? I think you might imagine that I want to say yes. Okay, so if you accept that there might be distinct specialties not rooted in incommensurable differences, then we can reach into what Mary Hess might call the neutral analogy and pull out a feature of biological speciation that could help clarify what's going on in Kuhn. So one of the long-standing questions in speciation is the role of biogeography. And here I got a quote from Kuhn and Orr, which is the sort of standard text in this. And the question of biogeography is, does speciation require complete geographical isolation, which would be called allopatric speciation, or can it occur without such isolation in populations that exchange genes, either to a limited extent, parapatric speciation, or freely, sympatric speciation? So you can see the sort of little picture of how these stories are different. In allopatric speciation, you only get a new species if there's this geographical barrier that allows them to develop independently. Whereas in parapatric, you have a new niche, and gradually different selective pressures can pull them into a distinct species, even though they are able to interbreed. And in sympatric, it's just, there's no niche. Just have an evolution of two distinct specializations. So if we bring this in from the neutral analogy, we can ask, are conceptual differences a crucial first step in the development of communicative barriers between specialties, or are there pathways of specialization and consequent communicative barriers that do not depend on these initial conceptual differences that Kuhn seems to be appealing to? And if I ask it that way, then I'm using incommensurable conceptual differences and as analogous to geographical isolation. Are there ways that you could speciate without it? That's what I'm looking at. Okay. Now, that was the breadth dimension. I'm going to go back to the depth dimension. So I'm going to recall it wasn't just that incommensurability is crucial to the proliferation of scientific specialties. It's also crucial to deeper and deeper investigation of nature within a paradigm or a scientific specialty. So we have a kind of story of how this works in structure. This is from early on. Focusing attention on a small range of relatively esoteric problems, the paradigm forces scientists to investigate some part of nature in detail and depth that otherwise would be unimaginable. So we've specialized. We've got a bunch of people that agree with me. So we focus on narrow problems. That's why we get such depth within a paradigm. No appeal to incommensurability at all in that. In late Kuhn, here's a continuation of his speciation metaphor. What permits the closer and closer match between a specialized practice and its world is much the same as what permits the closer and closer adaptation of a species to its biological niche. The increased inability of the residents of different niches to crossbreed, on the one hand, and the increased difficulty of communication between the practitioners of different specialties on the other. So somehow the fact that you can't talk to people in other fields is a crucial part of why you make progress within your own field. Just a little weird. A little weird to me, I guess. So why is that? Why would having incommensurable barriers with your neighbors be crucial to progress within your field? And the best I can sort of come up with is it's some analog to swamping. Like that, if your neighbors are, if you understand their ideas and are interested in their problems, you won't focus on your own or something like that. So somehow you'll be swamped by your neighbors if you're able to understand them or something like that. A couple questions about that. Like, if it's crucial that you don't understand your neighbors to do good science, why would it have to be conceptual barriers? Couldn't it just be, I haven't read the same journals, so I can't really follow what they're talking about? Wouldn't that work just as well? Anyway, so that seems to be the theory here. This should bring up a question to your mind, like, what about interdisciplinary science? What about all this progress in science? And this is a quote from Ladnovich. Modern science is unimaginable without numerous exchanges across theories, disciplines, and fields. Many scientific problems cannot be solved without the benefit of knowledge concerning other related aspects of the same domain. So it seems like interdisciplinary science is a crucial part of progress. He seems to be saying that not talking to your neighbors is crucial to scientific progress. How do we put those together into some kind of coherent story? Well, he does kind of mention what I'll call intersectional specialties. And he kind of has, in both structure and in later work, he pulls these aside as a kind of separate case. So in structure, he mentions biochemistry, and it's supposed to be a counterexample to his normal account of how sciences begin. So in the normal account in structure is you take any field of science and you go back in time and you go through a series of revolutions, and then there's this last step where you go to a proliferation of different schools, none of which is enough to form a paradigm. So out of these schools emerges the first paradigm. He says, that doesn't work for biochemistry. Why? Because it's at the intersections of fields that are already there and established, or something like that. So the story's going to be different. But he doesn't tell you the story. In later Kuhn, when he's talking about the speciation metaphor, he brings up these kind of cases again. He talks about cases when a new specialty has been born in an area of apparent overlap between two pre-existing specialties, as occurred, for example, in physical chemistry and molecular biology. What does he actually say about these? He wants to say, this is not a counterexample to my thesis that scientific progress occurs by specialization, because you don't get a new unified science when this happens. You just get a new specialty, biochemistry or molecular biology. This is a way that you get more specialties, not a way that science unifies over time. So that's what he wants to say about it. Yeah, it's not a counterexample because as time goes on, you notice that the new shoot seldom or never gets assimilated to either of its parents. Instead, soon you've got a department of biochemistry, journals of biochemistry, your own biochemistry curriculum. It's not assimilated to organic chemistry or biology. Okay. All right. So he doesn't tell us, but we might wonder, what's the role of incommensurability, if any, in the formation of these intersectional specialties, which would seem to require, again, intercourse between distinct specialties. So we've got interdisciplinary science and intersectional specialties, both of which appeal to some kind of interdisciplinarity and seem to put tension on this appeal to the essential role of incommensurability in Kuhn's account. Okay, so now we can go into some chemistry. So chemistry, as the kids these days might say, is a field with a high body count. That is to say, it's having intercourse all around itself with all the surrounding fields. So, and among the chemists, Linus Pauling is a notorious Lothario, right? He's learning learning physics and going over and contributing to structural biology. So chemistry is a natural place to look at cases of intersecting scientific fields or interdisciplinarity. So let me tell you a little bit about Pauling. He wins the Nobel Prize in he's actually one of, as Wayne reminded me, he's one of only 5 people to have won 2 Nobel Prizes. He won one in chemistry in 1954 and then one in the Peace Prize in the 60s. And he's one of only 2 that won them in different fields. Anyway, so 1954, he gets the Nobel Prize in chemistry. It's basically for this book, The Nature of the Chemical Bond, which he publishes originally in 1939. What is he doing in this book? Well, he sees himself as transforming structural chemistry from its classical to its modern form. So you might wonder, what does that mean? So structural chemistry is basically the idea that you describe the structure of chemical compounds, namely their bonds and maybe something about the distribution of their electrons, and you use that to explain their chemical and physical properties. So it's an approach to understanding chemical behavior in terms of structure. All right. Structural chemistry aspires to explain all this stuff in terms of structure. Now, chemists, structure it's a product of like the 1860s. And the notion, this strategy of explaining in terms of structure has persevered well until 1940, but there's been a lot of changes in what structure means over the course of that time. So originally it might be something like hooks and eyes, like somehow the atoms are hooked by hooks and eyes to other atoms. This is an early interpretation of what these chemical bonds might be. Eventually, someone discovers the electron. They're like, oh, no, it's shared electrons. So what structure amounts to evolves, but the strategy of explaining in terms of structure stays the same. And chemists are pretty open-minded. Like, you give me a suggestion about how to change my concept of structure and I can explain more with it, all good. Okay. So the strategy is preserved, but the actual content of what structure means changes. Well, Pauling initiates a change, another change in the conception of structure. It goes from basically, well, I guess I'll talk about it a little later on, from what he calls classical structure theory to modern structure theory. And it differs because of ideas that Pauling has imported from quantum mechanics. So he hears about the new developments in quantum mechanics. He gets a Guggenheim Fellowship, goes over to Germany for a couple of years, comes back, and then starts importing quantum mechanical ideas into chemistry. Okay, and this leads to this update to the concept of structure, which leads to a much more powerful structural organic chemistry. He gets a Nobel Prize for it. All right, also during this time, this book comes out in 39. Starting in the 30s, Pauling begins work on proteins. He ends up making a lot of important contributions to our basic understanding of proteins. He solidifies the macromolecular theory of proteins. Proteins are just big, long strings of connected amino acids. It wasn't clear. He offers a general account of biospecificity. That is how it is that one protein can bind to a particular compound, but not another. And then eventually he elucidates the major secondary structures, which are the sort of recurring structural features of proteins. And that's in the sort of early 50s. So he makes a lot of important contributions to the basics of protein science. So he looks like an interesting case for thinking about Kuhn because, one, he's made interdisciplinary contributions to both the depth of organic chemistry by bringing in physics, and then to the breadth of science in general by giving us the foundation of a new discipline, structural biochemistry. Okay, so that's why I picked him. Well, that's not the only reason. Okay, so classical structural theory as a reductionist research strategy. So this is just terminology from WIMSAT, which I think is kind of useful for thinking about structure theory. What classical structure theory does is it decomposes a complex system into its parts, analyzes them in isolation, and then resynthesizes these parts and the explanations of their behavior into a composite explanation. It's exactly what chemists do. They describe molecules in terms of recurring bonds and functional groups. You understand the behavior of the bonds and the functional groups, you can predict how a novel molecule behaves insofar as it's built of these same bonds and functional groups. So it's this decompose and reconstitute strategy for explaining the behavior of molecules. So classical structural theory does this. This is a picture of caffeine with basically a classical structural formula for it. So classical structural theory is this Lewis-Langmuir theory. The parts are chemical bonds between specific atoms, functional groups, asymmetric carbons, sometimes unshared electron pairs. That's what you use to describe a molecule in classical structural theory. You can explain lots of stuff using this, but even before Pauling, there are these sort of accumulating anomalies, cases where, based on the structure, you have trouble explaining its chemical or physical behavior. Not surprisingly, right? That's characteristic of reductionist research strategies. When you have a reductionist research strategy, they ignore, oversimplify, or otherwise underestimate the importance of the context of the system under study. You're trying to understand the system in terms of its internal parts, not so much the context that it's in, right? So that's a weakness of this kind of approach. So you can think about classic structural theory as limited by two kinds of context. One is sort of reaction context, like what a molecule do, its chemical behavior depends not just on its structure, but what it's in, like what solvent is it in, what temperature is it, what pressure is it at. These all affect the outcome of reactions. And over the course of the 20th century, chemists, physical chemists, develop all sorts of ways for accounting for structure, or sorry, for context in that sense. But there's also molecular context. So that is, not all double bonds behave the same. It kind of depends on what's around them, right? So if you're trying to build up the behavior of a complex molecule in terms of its parts, you can't just say it's additive. It depends what's nearby, right? So the molecular context of a functional group or feature influences its behavior. So in some way, you have to sort of account for the fact that, another way to put it might be, electrons are actually delocalized. They're not just in these little bonds that stay right there between the atoms. They smear out over the molecule. They affect each other. So you have to account for that somehow, and that's what I'm calling molecular context. Okay, so what does Pauling do? He introduces this concept of resonance. And a quote from him: "The theory of quantum mechanical resonance of molecules among several valence bond structures constitute a major addition to the classical structure theory of organic chemistry." So what is he doing? Oh, I guess I missed this last time. I forgot to talk about this one. This is a little picture of benzene, but it's written with alternating single and double bonds. And the point is that when you have a double bond that's adjacent to other double bonds like this, they don't behave like double bonds. They behave differently. They're called what are conjugated double bonds. So the behavior of benzene is not like that of a normal doubly bonded carbon atom. It behaves differently. The context affects its behavior. So now what he's going to do is explain why. What he says is, you don't just describe benzene like I did in the last page. Instead, you describe it like this. Everything in the brackets is now your formula for benzene. And you notice it has two Lewis structures in it, not just one. And that arrow in between them means that they resonate between each other, which basically, I guess, means that we should think of benzene as a sort of linear combination of these two, okay? You can't think of benzene as just being represented by this, and you can't think of it as just being represented by that. It's a kind of an averaging of the two of them. What does that mean? Well, that means, for instance, that this bond isn't a double bond and it isn't a single bond. It's more like a 1.5 bond. Okay. And where does that show up? Well, bond lengths are different. So the distance between the carbons in a benzene you would predict based on this is somewhere between the length of double bonds and the length of single bonds. That's in fact true. And then he also relates this to the energy of systems. So systems where you have this kind of resonating or lower in energy than either of the thing that's represented. So this is the idea. And what does this do? To some extent, it addresses the delocalization of electrons and extends, if you want to say that, the reductionist reach of structure theory. You can explain more stuff this way. You can explain why the properties of benzene are different from what's represented in this single structure. And yeah, okay. So it helps develop the concept of structure. You can explain a lot more cases. And basically, all you have to do is carry around multiple structural formulas instead of just one and kind of average over them at the end. All right, so what's important about it is philosophically, it's kind of interesting because it's certainly not a derivation of chemistry from quantum mechanics. The way Pauling describes what he's doing is using ideas from quantum mechanics to help develop some semi-empirical concepts for thinking about chemical structure. Okay, what are those semi-empirical structural principles that are, he says, compatible with quantum mechanics and supported by agreement with the facts of experiment? Some of these that you might have heard of: electronegativity, partial covalent bonds, hybrid orbitals, hydrogen bonding. These are all the terms that he develops by the semi-empirical method using concepts from quantum mechanics. He even acknowledges that resonance is somewhat arbitrary. And this is a reference to the fact that there are other ways of importing quantum mechanics into talking about chemical bonds that are different from resonance. So he's willing to acknowledge that. This is one way you might do this. But he says that's okay because the bonds that you started with were arbitrary too, right? Those are just some way of depicting the electrons around the atoms that's useful. That's what resonance is as well. So no big deal. Okay. All right, so now I'm going to show you how resonance helps. And I'm going to focus on two concepts that Pauling helps develop that actually get used in structural biology. So that's why I'm picking them. The first one is the concept of a hydrogen bond. In spite of its name, it's not a bond. It's a non-bonding interaction. So that just means it's a lot weaker than a typical covalent bond. A covalent bond is 90 kilocalories per mole, and this is like 5 kilocalories per mole. So these are weak interactions. They're first identified in sort of highly associated fluids like water. Water boils and melts at much higher temperatures than, say, hydrogen sulfide, so things that are in the same family on the periodic table. So somehow or another, water molecules hold on to each other much more tightly than hydrogen sulfide. So you want to explain, how is it that water molecules hold on to each other so tightly? And this is the basic answer that you have a picture up here. The red ones are hydrogens, the blue ones are oxygens. And we see that there's a partial positive charge on the hydrogen, a partial negative on the oxygen. And so there's some electrostatic attraction between them. That accounts for the hydrogen bond. And so these occur when hydrogens are bonded to electronegative atoms, NO and F, basically. And then they can interact with another electronegative site. So what you want, if you're Pauling, is a structural explanation. Why is it that water does this? Well, he says, normally we think of water in this way as just two hydrogens covalently bonded to an oxygen. But that's not an accurate representation. Really, we need all four of these Lewis structures to talk about water. It's not a pure covalent bond. This is an ionic bond between oxygen and one hydrogen and a covalent with the other. This is a covalent with this one and an ionic with that one. And this is a double ionic bond. So really, water is, if you want, a kind of linear combination of these four representations. And what that means, you'll notice, is that in these other representations, the oxygens have a lot of negative on them. The hydrogens have a bunch of positive. So the actual water molecule has what you would call partial negative charge on the oxygen, partial positive on the hydrogens. So that's a better representation of water than the standard one. And once you see that, you'll say, oh, it's going to have a dipole. Dipoles interact. That's why water sticks to each other. What? Yeah, that's his estimate of the relative contributions of these various Lewis structures. And he has complex ways of figuring that out. Okay, so OH bonds have what's called partial ionic character. They're a mix between covalent bonds and ionic bonds. And so they have charge separation, and so they interact with each other. Electronegativity, which I used up here, is another one of these semi-empirical concepts that he introduces that's basically supposed to measure how hard does a particular element pull on the electrons in its covalent bonds. So when something is more electronegative, it's greedy. It pulls the electrons towards itself. So oxygen is greedy, pulls those electrons towards itself. It's got a large partial negative charge. So this is a numerical scale which he develops. Okay, so the upshot is that now you can look at a chemical formula and say, aha, there's the potential for hydrogen bonding here because I've got a hydrogen bonded to an oxygen. And oh, there's another oxygen over here. So this molecule could potentially form hydrogen bonds with itself or with other molecules. It's an identifiable structural feature that's there in the chemical formulas once you know about electronegativity. Okay. And then here's a quote from him about this. I believe that as the methods of structural chemistry are further applied to physiological problems, it will be found that the significance of the hydrogen bond for physiology is greater than any other single structural feature. So he thinks that hydrogen bonds are the key to it all. Well, here's another important concept that he develops that gets used in the study of proteins, and that's what he calls partial double bond character. And this is, I guess, relatively easy to see with a peptide bond. So the peptide bond are what link together amino acid residues in a protein. And it's this bond between nitrogen and carbon, where the carbon has a double bond down to oxygen. Well, Pauling says, no, you can't just represent a peptide bond this way. You'll miss out on features of the peptide bond. It's really got to be represented as a sort of linear combination of these two. And why that's significant is that in this side, we've got a double bond between the carbon and the nitrogen. And double bonds are importantly different from single bonds in a couple ways. One, they're shorter. And two, there's not free rotation about them. They're locked in. So it takes a lot of energy to break a double bond and switch the direction of the things on this side of it. So peptide bond can't be represented with one Lewis structure. The CN bond is single in one, double in the other. The actual molecule is a sort of, it's 40% the double bond and 60% the other one. So you expect that there should be a 40% double bond rotational barrier around the carbon-nitrogen bond. In physiological conditions, that's insurmountable. So you're basically locked into a plane on the carbon-nitrogen in the peptide bond. Okay, this, he says, has been of much value in the determination of the structure of proteins. Basically, what it does is if you look in this protein, in this residue, you've got three bonds, the CC bond, the CN bond, and the NC bond. Presumably, you could rotate around any of those three bonds, so the potential space for a protein chain would have 3 dimensions of freedom. If one of them's locked, you've reduced that to 2 dimensions of freedom. And that's why when you see these Ramachandran plots, they're only in 2 dimensions because one of those bonds is locked. That's what Pauling's realizing with the partial double bond here. So it basically reduces the degrees of freedom in the description of the protein chain. And the consequences on the basis of the theory of resonance, the planarity of the amide group greatly reduces possible structures of a protein. Okay. All right. So what does he do with all this? Well, this is the last paragraph of the nature of the chemical bond. Among the most interesting problems of science are those of the structure and properties of substances of biological importance. I have little doubt that in this field, resonance and the hydrogen bond are of great significance, and that these two structural features will play an important part in physiological phenomena. Maybe many years before we understand molecular structure well enough to explain proteins with highly specific properties, such as those shown by antibodies, which must be attributed to their possession of well-defined and complex molecular structures. But the attack on these substances by the methods of modern structural chemistry can begin now. Ultimately, it will succeed. So to some extent, he's right, and he does some of the work. So let me show you what he actually does. And to do that, I would talk a little bit about where protein science was before Pauling. So by about 1930, there was a broad consensus that when you denatured a protein, it was a polypeptide chain. So denatured means taking an egg and then cooking it. So after you've cooked it, it no longer functions like it's supposed to. It's now this big mess of congealed mess, right? That's denaturing the protein. Same with an enzyme. If you have an enzyme and you heat it up or you put in chemicals, it won't work anymore. It won't catalyze the reaction it's supposed to catalyze. So denaturing is taking something that's biologically active and messing it up so it's no longer biologically active. And you can do that with heat or chemicals. All right, So they knew that once you denatured them, they were polypeptides. But what they didn't know was what's the structure of the active forms of the protein. And so there were theories going around that were something like this. In order to explain the specific biological and physical properties that proteins lose when you cook them, it's necessary to assume the presence in the active proteins of covalent linkages other than the peptide bond. So they're thinking maybe it's more complicated, much more complicated than a chain of amino acids. Maybe there are all these links between the different parts of the chain. One of them, the cyclol theory, imagined that there were 4 connections between each amino acid residue, not 2. Why? Because then you could get these elaborate cages, which would only let in certain shaped molecules or something that was going to explain biological specificity. And in order to do that, you needed a much more rigidly defined structure in the active state of the protein. So what does this mean? When he comes on the scene, the macromolecular theory needs an account of what happens during denaturation and how you can explain biological specificity if all you've got is a protein chain. Okay. Well, that's what he does. So in 1936, Pauling publishes his own account of what's going on during denaturation. In 1939, he publishes this sort of critique of the cyclol theory, saying basically it's energetically impossible. Plus, you don't need it. Plus, it doesn't match with the X-ray crystallography. So he's destroying the alternative accounts of proteins that are on the scene. And he provides here in 1936 basically our current conception of what happens during denaturation. So our conception of a native protein molecule is the following. It consists of one polypeptide chain, which continues without interruption throughout the molecule, or sometimes chains are linked together. This chain is folded into a uniquely defined configuration, which is held by hydrogen bonds. The denatured protein molecule we consider to be characterized by the absence of a unique configuration. As a result of increases in temperature or reagents, the side chain hydrogen bonds are broken, leaving the molecule free to wiggle around in any large number of ways, which is the congealed protein. So this is basically a modern conception. He acknowledges other non-bonding interactions besides the hydrogen bond. But it's actually 20 years after all this before anyone identifies what are now centrally important hydrophobic forces. So that's only later on. But hydrogen bonding and all the others that you would normally think of, Pauling articulates, but not hydrophobic forces. So collectively, he says, interactions of this type, yeah, they're weak, but collectively, they're enough to stabilize the structure of a protein. We can account for a consistent structure of a protein just by appealing to these non-bonding forces. Okay. What else does he explain? Biological specificity. What does this refer to? So a protein or a particular enzyme will only catalyze one particular reaction. It's very specific about which other chemicals it will actually interact with. It's specific. Not an antigen. An antibody will only react with a particular antigen. That's another case of biological specificity. So it's a puzzle. How is it that these biological molecules are so selective about what they interact with? Well, he's got a story about that too. The weak non-bonding forces between molecules are such as to give stability to a system of 2 molecules with complementary structures and juxtaposition. Complementariness should be given primary consideration in the discussion of the specific attraction between molecules and in the enzymatic synthesis of molecules. So he's got this story. It's complementarity, which explains the biospecificity. And then this is just a summary from a historian of biochemistry. The notions of weak bonds and structural complementarity are still the basis of our understanding of macromolecular interactions, and these principles govern the structure and function of all forms of life. And this is a picture of 2 complementary base pairs in DNA, which in fact Pauling missed. He worked on the DNA problem, but he didn't notice this complementarity in the base pairs. But anyway, the idea of complementarity is still very, very much alive. Okay, one last contribution, and that's secondary structure. This is later in his career. There are these X-rays of keratin or stretched hair going around that have this kind of repeating pattern. And people are trying to figure out what could the possible structure of a protein be that would explain this repeating pattern that we're getting in the X-ray crystallography. Pauling approaches it in the following way. He says, well, I know all the bond distances in amino acids and small peptides, all the carbon, nitrogen distances. I know where the hydrogen bonds are likely to line up in a dipeptide. Let me just extrapolate that to really big proteins. And so the constraints he puts on his model are the CO group from each amino acid residue accepts an NH hydrogen bond from another residue. So those are the yellow lines here. And all 6 atoms are in a plane. That is, the amide bond is flat, like we got from the partial double bond character. And he says on those constraints, the only way we can wind this chain around and have all these hydrogen bonds and keep the plane flat are these 3 ways. The left-handed alpha helix, the right-handed alpha helix, and the beta sheet. So he publishes these 3 ways that you can maintain all these hydrogen bonds and keep the peptide bond flat in 1950. And then 10 years later, when they do X-ray crystallography of myoglobin, it's 85% alpha helix. Beta sheet also occurs throughout proteins, but their mix sort of depends on the protein. But he started this kind of novel bottom-up model building approach to addressing the structure of proteins. This also, of course, gets applied eventually to DNA. Pauling doesn't succeed there for interesting reasons, which is that he forgot that the phosphate groups will lose their H's at biological conditions. So it was the conditions which got him wrong. Being an organic chemist, that makes sense. But anyway, so this model building approach is eventually applied to DNA successfully, and it generalizes to the sort of protein folding problem that you have now. Okay, so these are Pauling's contributions to organic chemistry and the birth of biochemistry. So now we can go back to the Kuhn and see what we can say about his accounts of the role of incommensurability in scientific progress. So recall that along the depth dimension, incommensurability was supposed to prevent swamping from neighboring disciplines and thus allow a, quote, closer match between a practice and its world. So in this case, I'm thinking about what happens with Pauling bringing quantum mechanics into organic chemistry. Is incommensurability preventing swamping by neighboring disciplines? It just seems like the completely the wrong direction to take for the way that progress occurred in this particular case. In fact, it seems to depend on some kind of intercourse with neighboring disciplines, the one that was funded by the Guggenheim Foundation when Pauling went over to study with these physicists. By moving structure, I have a count. By moving structure theory from its classical to its modern form, Pauling does allow for better explanation, predictions, and applications in organic chemistry. So there is an increase in the depth of organic chemistry if you want. But it's not, I think, by preventing its interaction with physics. Instead, a different kind of broadly Kuhnian story seems plausible. That is, it's an articulation of the concept of structure using a novel interfield theory, the theory of resonance, that connects it up with a neighboring discipline. Resonance allows for a natural extension of the concept of structure. That's why it's an articulation, not a revolution, I guess, which preserves the strategy of structural theory, which is to build up the behavior of complex molecules in terms of structurally identifiable features. Preserves that, but makes it better at its job, basically. Again, though, I don't think it's a case where a discipline progresses by getting less specialized. So it's not really a counterexample to the case that Kuhn introduces, that like science gets better by getting more and more specialized. It seems like that. It's just that it gets more specialized by co-opting ideas from an adjacent discipline and refining its approach using those ideas. So this preserves, I want to say, Kuhn's original insight and structure that an increasingly articulated paradigm is central to scientific progress along the depth dimension. But it's compatible with a robust role for interdisciplinary incourse, and it doesn't require appeal to incommensurability to prevent swamping. So you can get a roughly Kuhnian account of how you would get progress within a discipline with help from enabling discipline that doesn't have anything to do with incommensurability. What about the proliferation of specialties? So now I'm thinking about the kind of birth of biochemistry case. So go back to what Kuhn had to say about this. Incommensurability is supposed to lead to the proliferation of specialties because there's some kind of initial conceptual incongruity which is introduced to help solve anomalies in the ongoing practice. And this forces the gradual development of incommensurable taxonomies. To solve these problems, you need to change your categories, and the rest of the discipline keeps going on in its own way. So you split the discipline into distinct disciplines that have restricted intercourse. And again, I just don't think this fits with what Pauling's doing in the use of structural organic chemistry to explain biological phenomena. Instead, Pauling's big insight is that biomolecules are really just really big organic molecules. They obey all the same principles as their smaller, more synthetically manageable cousins. So it's the continuity between structural organic chemistry and structural biology that was the key to its success, not any kind of incongruity. Instead, I see it like this. Pauling's work on the chemical bond expands the explanatory power of structural organic chemistry. It gives all these new concepts, partial double bonds, electronegativity, hydrogen bonding. And using these concepts, all of a sudden there are questions in biology that become accessible to structural organic chemistry, can explain things that they couldn't explain before. With these tools, modern structural theory allows a solution of outstanding biological problems. What is denaturation? How is biological specificity possible? And a program for predicting the conformations of proteins. That's the alpha helix beta sheet work. It's not, again, however, I think a counterexample to the idea that science progresses by proliferation of specialties, because in fact, structural biochemistry does become a distinct specialty in spite of its conceptual continuity with structural organic chemistry. So how would that work? We tell a story about how, in spite of the conceptual continuity with organic chemistry, structural biochemistry becomes its own field, which it decidedly is. It has its own journals, it has its own departments, you learn different things. After a while, at least on the details, you might have trouble understanding each other even. Well, I think we can think about it as an articulation of structure in organic chemistry opens up a new problem-solving niche. That is explaining denaturation or the structure of proteins or biological specificity. But the selection pressures within this niche are different from those in mainstream organic chemistry. The kinds of problems you're solving are pretty different, right? And mainstream organic chemistry, you're trying to synthesize novel compounds. The problem is a synthetic problem. In structural biochemistry, this is a little bit reductive, but you're trying to give a structural explanation of a biological function or something like that. Very different problems that you're starting with. And those problems lead to different explanatory focuses. In organic chemistry, it's all about the covalent bonds. Of course, non-bonding interactions are important, et cetera, but really you're concerned with covalent bonding. And in structural biochemistry, the non-bonding interactions are where most of the action is. Okay, so it's not that they're incommensurable, which is they're focusing on different parts of the organic molecule. There's also an important different relationship to their context. So for an organic chemist, the reaction conditions are a variable that you can adjust to help you do what you want to do. You can change the solvent, you can change the temperature, you can change the pressure, and you might do that to help get the reaction go the way that you want. In the context of structural biochemistry, it's a parameter that it's fixed by the biological conditions, right? You're trying to understand how things behave in these particular conditions. And so you want to, you're directed to a particular set of conditions. This means that there are different kind of characteristic challenges. Synthetic design is the characteristic challenge of organic chemistry, and the in vivo in vitro problem is a characteristic challenge for biochemistry. Like, I can understand what's going on with these things once I've changed their chemical conditions, but how do I project back into what's going on in biological conditions? So basically, you've got different problems. They lead to different focuses and different characteristic concerns. And so we have a version of parapatric specialization, not allopatric specialization. Even though there aren't incommensurable conceptual barriers, specialists in the two fields need to know different things. They're solving different kinds of problems, using different examples, different strategies, different instruments, and eventually subgroups diverge, communication barriers develop. We've got specialization without incommensurability. Okay, so what does this all do for me? I want to say incommensurability isn't necessary for progress along either the depth or the breadth dimension. He should just drop this whole swamping thing, and he should broaden the speciation metaphor. That depended on this idea that specialization itself can be the cause of communicated barriers, which he introduces in structure, and it's not always the effect of incommensurability, which he seems to say in later work. Dropping swamping allows for a natural account of interdisciplinary progress within a specialty, which I described, theory articulation aided by an interfield theory. And the broader version of the speciation metaphor has better prospects for accommodating intersectional disciplines because it allows specialization without conceptual incommensurability, parapatric specialization, if you want. So what does Kuhn have to give up for all this? Well, the essential role for incommensurability in scientific progress. If only if he pulled back on that incommensurability mission creep, he would have had a much more generally plausible and broadly applicable account of progress that preserves the original insights and structure. That's it.